Welcome everybody to our webinar today. I am Elisa Nent, the Product Manager for Neuroscience at Miltenyi Biotec. And today you will learn about cell sorting for the enrichment of neuronal subpopulations, new perspectives for clinical research, which is given by Dr. Thomas Andreska of the Institute for Clinical Neurobiology in Würzburg. Before we start, I would kindly present to you today's agenda. So I will begin with a brief introduction about our neuroscience portfolio. Then I hand over to my colleague Shenjun Jin, who is the product manager for the MACSQuant Tyto cell sorter. After this initial part, we are then coming to our main speaker, Dr. Thomas Andreska, who's going to explain us his recent project about the sorting and cultivation of neuronal subpopulations. I would kindly ask you to post your questions into the chat, and then afterwards we will have some time to address them. So now let's start with the neuroscience portfolio. And here this image represents our two main workflows that we established for your neuroscience research. So on the left-hand side, you can see the clearing and the 3D imaging workflow to acquire spatial information of the brain. And on the right side, you see the dissociation, separation and analysis workflow for the in-depth analysis of cells. So for today, let's only focus on the right workflow. This workflow goes from sample preparation over to cell separation and can be followed by cell cultivation or analysis. Depending on the age of the animal, we recommend the neural tissue dissociation kit for neonatal samples or our adult brain dissociation kit for the adult brain. And we have more products for the cell isolation and analysis that I will present to you further in the following slides. Okay, let's start with sample preparation. The GentleMax octodissociator with heaters helps you with a standardized and automated dissociation of the brain tissue. It uses mechanical disruption and enzymatic digestion for a very gentle and efficient brain dissociation. Especially for the adult brain, we developed the Adult Brain Association Kit, which includes a cleaning step to remove all the cell debris and the red blood cells that would normally hinder any further cell separation. This kit results in a high yield of pure and viable cells, which are then ready for many downstream applications, such as single-cell sequencing, for example. After the sample cleaning, you are able to isolate your neural cells with a variety of our microbeads. This magnetic cell separation technology is a very gentle and fast way to obtain your cells of interest out of the cell suspension. It takes less than an hour and will result in a high purity and viability. And to reduce your hands-on time, we also offer the Automax Pro separator, which can be used to automate the cell separation process, meaning less work for you. The cells that are isolated from our workflow are then, as I said, very viable, therefore can be even cultured afterwards. We offer serum-free cell culture media and optimized supplements to cultivate all kinds of neural cells. And the serum-free formula prevents the variability that normally would appear using different FCS slots. If you are working with PSC-derived neural cells, we have this optimized dMMax PSC Pro XF media that allows for weekend free feeding. And to boost your cultured cells, we offer a lot of high-quality cytokines for many different applications as well. If you then want to analyze your neural cells, we offer a variety of different antibodies for neural cell applications, which are suitable for IF stainings, for example, as well as for flow cytometry. For the analysis of your cells, we recommend you the MACSQuant analyzer. But in case that you are more interested in isolating cells based on more than one marker, and for this the isolation with our microbeads wouldn't be sufficient, we can recommend you the MACSQuant Tyto cell sorter because this instrument provides a very gentle and sterile way of sorting, and this is specifically helpful for the sensitive neural cells. But before I go too much into detail here, I would now hand over to my colleague Shenjun Jin, who is much more competent in explaining you all the advantages that the Tyto has.
Thank you, Elisa, for your introduction. Hello everyone. My name is Chengjun Jin. I'm product manager for MACSQuant Tyto self sorter. In my part, I would like to introduce you the unique technologies of our instrument and tell you why we think Tyto is a good option for sorting fragile and sensitive cell types such as neural cells for the important downstream applications. MACSQuant Tyto is a closed and self-contained benchtop cell sorter. It is quite compact and it does not require any fluidics in the instrument. With a large external monitor, it is easy to analyze data and implement a gating strategy. A disposable Tyto cartridge holds a sample and is housed in a closed and temperature-controlled chamber where the sample can be kept at a constant temperature from 4 to 25 degrees Celsius. There are 3 lasers available, with 8 fluorescent channels and 2 scattered channels, which provide full experimental flexibility and the possibility of multi-parameter sorting. This is the MACSQuant Tyto cartridge and the real heart of the Tyto system. There are 3 chambers: the input chamber for the sample, the positive chamber for the target cells, and the negative chamber for all negative cells. Different from the conventional droplet cell sorters, the MACSQuant Tyto has no fluidic path inside the instrument. It means the cells do not run through the instrument itself. Instead, prior to sorting, cells are loaded to the cartridge and will still remain in the cartridge after the sorting. And because the cartridge is a single-use consumable, there are no sample-to-sample carryover between runs. When viewing from the bottom, you can see the small microchip where the sort process really takes place. And this is a real tiny part. As you can see, this is much smaller than match end. Let's take a closer look at the sorting process more in detail. The cells from the input chamber go through the sorting channel in the microchip and through the laser beams. So once the target cells were detected by the system, a magnetic pulse will just open the microvalve so that the target cells will be then sorted into the positive chamber, and all the negative cells will then go into the negative chamber in the end. Let me show you a short video for this process. As you can see, the valve opening and closing, and now there are cells that are coming from the positive chamber, go through this microchannel. And as you can see here, the green target cells are being recognized by the system and collected into the positive chamber, and the microvalve will quickly close again so that all the negative cells are moved forward into the negative chamber. Why did we decide to push the idea of a cartridge and microvalve-based self-sorter? Because there are so many problems associated with the conventional droplet self-sorter. First of all, the conventional self-sorting is very harsh to cells. The cells are sorted under high pressures, strong shear forces, and the cells will be decompressed and charged during the sorting. All these factors can negatively affect the viability and functionality of cells. In addition, due to the shared fluidics and tubings inside the instrument, there will be sample-to-sample carryover and contamination issues. And all the droplet self-sorters have open sorting system, meaning there is a huge safety concerns, and they have to be used under a huge biosafety cabinets, especially for sorting infectious materials. Since 2020, sorter-induced cell stress, also called SICS, has attracted increasing attention and been widely discussed all over the world. If you are interested in this topic, please have a look at this publication. In contrast, when comparing our Tyto sorting principle with conventional droplet cell sorters, there are clear differences. Tyto is gentle self-sorter, and cells enter the microchannel under extremely low pressure. Cells do not get decompressed, and no charge is applied on the cells during sorting. And as I already mentioned, because the Tyto cartridge is a single-use consumable, there are no sample-to-sample carryover and contamination problems between runs. In addition, the sorting is happening in a completely closed environment on the Tyto, so there will be no aerosols generated during sorting, which is also very important for operators' safety in the lab as well. Due to the time limit, I cannot explain too much in detail. If you are interested in our instrument and need more information about Tyto, please visit our website. Now I'm glad to introduce our speaker, Dr. Thomas Andriska. Dr. Andriska is now working as a research scientist in the lab of Professor Dr. Santner at the Institute for Clinical Neurobiology at the University of Würzburg. He is also the project manager for Parkinson's and dysthymia in the German National Research Project SFB-TRR295. Now I would like to hand over to Dr. Andriska, and I'm looking forward to hearing more about his exciting research.
Hello and a warm welcome to this webinar series. And of course, a big thanks to the organizing committee for getting the chance to present some of our data here. My name is Thomas Andreska. I am a postdoc in the lab of Professor Michael Sentner at the Institute of Clinical Neurobiology in Würzburg. And I'm going to talk about cell sorting for enrichment of neuronal subpopulations, which opens new perspectives for clinical research. Now we are interested in the basal ganglia circuit, which is involved into voluntary movement control and movement initiation. And of course, this is important for Parkinson's disease. In particular, we are interested in the communication between the somatomotor areas in the cortex and the caudate putamen in the dorsal striatum. So this part of the striatum is the main region involved into movement control and does not only receive glutamatergic input from the cortex, but also dopaminergic input from the substantia nigra in the midbrain. So if we take a closer look into the striatum at the synaptic connection, we are focusing on corticostriatal synapses, which is the physical interface actually for information processing between dopaminergic fibers from the midbrain and glutamatergic fibers from the cortex, which target spines on postsynaptic medium spiny neurons in the striatum. And these synaptic connections are not static but very dynamic and require structural and functional plasticity in order to learn and modify motor skills. And the molecular basis for this plasticity is basically long-term potentiation or long-term depression. And there's one factor that is critically involved in those processes, and this is brain-derived neurotrophic factor. And together with its postsynaptically expressed receptor TrkB, it modulates a neuronal survival, development, but also synaptic plasticity by modification or modulation of LTP and LTD. So we were wondering if neurotrophin signaling at these synapses is actually altered in Parkinson's disease due to the absence of dopamine. And basically, one would suggest in order to investigate this, we could dissect the striatum. In this case, of course, we would find astrocytes, then neurons, the vast majority of neurons in the striatum with 95% approximately are striatal medium spiny neurons and 5% of interneurons. And as a next step, one would obtain a single cell suspension, set up a primary medium spiny neuron culture and plate those cells, and then just investigate activation of dopamine receptors in those cultures and investigate the effects on BDNF TrkB signaling and interaction. The only problem is it's basically not that easy. Because if we take a look at those medium spiny neurons, first of all, we find two subpopulations. The first one is expressing dopamine receptor 1. And these medium spiny neurons are called direct pathway medium spiny neurons because they directly project from the striatum to the internal segment of the globus pallidus or the substantia nigra. The other subpopulation is called indirect pathway, iMSNs. They express dopamine receptor 2 and they indirectly innervate the internal segment of the globus pallidus through passing the external segment. So all the projections of those neurons already end in the external segment of the globus pallidus. And both subpopulations in common have the expression of dopamine and cyclic AMP regulated phosphoprotein of 32 kilodalton, simply called DARP32. So this is a common marker to identify both types of medium spiny neurons. So the first problem that we had was how to distinguish the direct pathway medium spiny neurons from the indirect pathway. And as you can already see, this just works so far with transgenic mice. So to identify direct pathway medium spiny neurons, we used these dopamine receptor 1 tandem tomato mice. They express the fluorescent dye tomato under control of dopamine receptor 1 promoter in striatal direct pathway. And if we then dissect the striatum of those mice and set up a primary culture without sorting, it looks like this. So we can see quite a lot of nuclei. The larger ones you can already suggest that these are astrocytes. We can see the MSN marker expression DARP32, which is expressed in both subtypes, and we can see single tomato positive medium spiny neurons of the direct pathway. So the only problem that we have in those cultures now is that the direct pathway neurons only represent 50% of all medium spiny neurons because we have a mixture of direct and indirect pathway. Furthermore, we will also find some, but very few, interneurons. And the biggest problem that we have, as already mentioned, we have astrocytes in those cultures, and astrocytes are dividing, and over time they will overgrow our neurons. To identify the indirect pathway medium spiny neurons, we used dopamine receptor 2 EGFP mice. And again, if we set up a primary non-sorted culture, we can see lots of astrocytes. We can find our medium spiny neurons, of which the indirect pathway medium spiny neurons now express EGFP. There are some few interneurons, and again, the vast majority are astrocytes. So the aim would be to specifically identify and purify those medium spiny neurons. And when we quantify the amount or the concentration of medium spiny neurons in those non-sorted cultures, we will end up between 2 and 5% DARP32 positive medium spiny neurons of both subtypes. This also means that the direct pathway represents only 1 to 2.5%, as well as the indirect pathway. So the concentration is very low, and the problem is how will you investigate subtype specific effects with such a low amount of neurons. So our aim was to increase the concentration of not only all medium spiny neurons, but specifically subtype specific medium spiny neurons to approximately 80%. And how will you do this? And when I came up with this idea, it was in 2013. I was a young PhD student at that time, not so much experience actually. I had the idea, if we have such nice transgenic mouse models, we could use a FACS sorter to purify those neurons from our single cell suspension. And so to say, purify the direct pathway from the tomato mice and the indirect pathway neurons from the GFP mice, and just sort out the astrocytes and interneurons which go to the waste. And I thought it's a pretty good idea, and my supervisors somehow didn't really agree, as you might imagine, because I didn't know so much about FACS sorting. I just thought it's an interesting technique that enables actually purification of those neurons. But of course, we have several problems. So first of all, setting up a single cell suspension after dissecting the striatum, trypsinization and so on, standard procedure, takes quite a lot of time. The next problem is that these cells are then shot through hoses and through a tiny little nozzle at the FACS sorter at a certain pressure. So the cells are then physically stressed. Followed then by getting grilled in a laser due to the detection of the fluorescent dyes. And the biggest problem is then getting charges on the cell surface and getting sorted in a high voltage electric field, which is not very good for electrosensitive cells like neurons. And at the end of the day, I was told all neurons will die, at least within 2 days. However, I thought we hit the first two important criteria. We have a specific marker expression for target neurons. And I really have to point out this. This is probably the most important point. We really need specific marker expression in our desired target cell type. We are not sorting neurons, we are sorting a fluorescence, and it depends on the specificity of this fluorescence in our desired cell type to end up with a highly pure and good sort. So we need at least a specific fluorescence or a biomarker, for example, on the cell surface to specifically identify our desired cell type. And the second criterion was the reliable detection with the FACS sorter. So you can see here, I included a negative control without fluorescent marker expression, which is clean. And from this, we can clearly identify then our positive population of direct pathway neurons in this case, or indirect pathway GFP expressing neurons in this case. I performed the first sorts actually, and I still had in mind that my supervisors told me they will survive for maybe 2 days and then they will all die. At least I got the chance to do those sorts and to give it a try. And when we made the first sorts, I thought after 24 hours, cells look pretty good. As you can see, they start to form neurites. Even after 48 hours, they still look good. But then my supervisors turned out to be right, because after 5 days, they started to look horrible. Even worse at 7 days and so on. And basically after 10 days, all my cells died. But at least it was already 7 to 10 days. So we found that the sorting parameters are very important because the cells are very sensitive to those parameters like laser power, pressure, electric charge, and plate voltage. And one has really to take care of decreasing the time between the dissection and getting in contact with the cell culture and ending up in the incubator. So one really has to hurry up. And temperature, of course, is also important at the time of the, during the whole time of the procedure. And when we adjusted these settings, took us approximately 2 months, the cells started to look better. So far, after 2 days, not much of a big difference, but then afterwards, 5 days in vitro, they look pretty healthy. They form nice networks. At 7 days, they were still alive. At 10 days, they were forming denser networks. And to see if during this development in culture cells died, I photographed the same area time and time again. And with those magenta arrows, I labeled cells that are dying over the time, but still the other neurons are nicely developing, they are staying alive and actually look pretty good. Overall, it was not that difficult to keep them alive for at least 14 days, as you can see over here. And the longest culture I ever had was like 21 days, but I never performed experiments after 21 days in vitro, so I always stopped at that time. And I never tried how long I could actually keep them alive in cell culture. However, when it came to the concentration, in this case of indirect pathway medium spiny neurons, at 14 days in vitro, we could show that 80% of the cells in those cultures express DARP32, which is the general marker for medium spiny neurons, and approximately the same amount expresses GFP. And slightly less, like 75%, of those cells in those cultures are DARP32 and GFP double positive. So meaning that at 14 days in vitro, purification of indirect pathway medium spiny neurons ends up with a concentration of approximately 75% of indirect pathway medium spiny neurons in those cultures. And still after 21 days, we still ended up with approximately 60% indirect pathway medium spiny neurons in those cultures. So, of course, the culture conditions are also important, meaning cell density, presence or absence of serum, serum concentration, trophic support, etc. The next step was then to not only identify these neurons by like DARP32 expression or GFP, but specific markers. And I just want to quickly present our data on the indirect pathway medium spiny neurons. Of course, they express DARP32. They are GABAergic like all medium spiny neurons. They specifically express dopamine receptor 2 and enkephalin, a marker for dense core vesicles. And when we stain those cells, as you can see over here, in green is shown, of course, the GFP expression, and in red, DARP32. So, as mentioned before, the amount of DARP32 GFP double positive neurons is very high, as you can also see over here. So these cultures are sorted and kept for 14 days in culture. That's what they look like then. And so DARP32 expression is clearly there. Next, we checked for GABAergic markers, therefore we used GAT65. And as you can see, there's a dense expression of GAT65, and one can also see like these boutons or synapse-like structures on the surface of those neurons, indicating already that they form synaptic connections between each other. Then we also checked for enkephalin, which is the first specific marker for indirect pathway medium spiny neurons, a dense core vesicle marker, and indeed it looked like dense core vesicles and enkephalin expression was also present in all GFP positive neurons. And finally, we wanted to investigate dopamine receptor 2 expression, and since there's not really a specific marker for dopamine receptors because they're quite conserved, we tried to detect dopamine receptor 2 mRNA, and indeed we could show that dopamine receptor 2 mRNA is present in those GFP sorted indirect pathway medium spiny neurons which express DARP32, while direct pathway would express dopamine receptor 1. We were not able to detect dopamine receptor 1 mRNA in those cases. All right, so in summary, in case of the direct pathway, we started with a primary non-sorted culture with approximately 5% of all medium spiny neurons, and after sorting those neurons, we obtained FACS-enriched direct pathway medium spiny neurons at a concentration of approximately 60%. In case of the indirect pathway, also we started with 5% medium spiny neurons in non-sorted condition and 70 to 80% indirect pathway medium spiny neurons after sorting. So the question is now, why is there such a difference? And I quickly want to explain the importance of the specificity, again, of the marker expression. The problem that we had with the dopamine receptor 1 promoter, it's not only active in striatal direct pathway medium spiny neurons, but as you can see, it's also active in lower layer cortical neurons, and to some extent it's also active in astrocytes in the striatum. Meaning, we actually, as I said before, we do not sort these direct pathway medium spiny neurons. We sort for the fluorescence of tomato, and that's present if we do not clearly dissect the striatum. We might get cortical contamination, and still we have weaker but still present tomato expression also in striatal astrocytes. So in some cases we might get some astrocytes also into those cultures. They will divide, grow, and then will of course decrease the concentration of our direct pathway medium spiny neurons. Alright, so now what's the advantage of this sorting mechanism or this sorted cultures? So coming back to what I initially presented, we're interested in the interaction between BDNF and TrkB. TrkB, which is expressed in those striatal medium spiny neurons, and actually there are two subtypes of TrkB. One is the 140 kilodalton sized full-length TrkB, which is more or less specifically expressed in neurons, versus the truncated TrkB receptor with a size of or molecular weight of 110 kilodalton. This one lacks the C-terminal signaling domain, and this is predominantly expressed in astrocytes. And when we compared non-FACS sorted to FACS sorted cultures in terms of Western blot analysis, we first of all ensure that all lanes are equally loaded by calnexin, and then we compared TrkB expression in those cultures. So in non-FACS sorted cultures, we can find a significant band at 110 kilodalton, representing the truncated receptor, due to the high amount of astrocytes, and we cannot really see 140 kilodalton full-length TrkB. After FACS sorting, that looks a bit different, so we significantly decrease the amount of the truncated TrkB receptor and increase the amount of full-length TrkB by increasing the amount of striatal medium spiny neurons. So we can now detect this receptor that was not visible before and investigate effects on this receptor like phosphorylation and so on. Next point in making good arguments, we could not really detect the medium spiny neuron marker DARP32 in non-sorted cultures, which makes sense because we only have like 5% medium spiny neurons. But after sorting, we can clearly detect DARP32 due to the high amount of medium spiny neurons. And finally, to see functional effects, we stimulated those cultures with the dopamine receptor 2 agonist sumanerol, because we wanted to know if there are effects on BDNF TrkB interaction after activating dopamine receptor 2 in this case. So we had to ensure that our agonist indeed activates dopamine receptor 2, and it was published before that this activation will increase phosphomap kinase between 30 seconds and 15 minutes, which is barely visible in non-sorted cultures. So one could say, okay, here after 15 minutes we can see an increase in phosphomap kinase compared to the FACS sorted cultures, where we can clearly see this described increase after 30 seconds and 15 minutes, and also described in the literature that the phosphomap kinase levels will return to baseline and below after 60 minutes. And compared to total MAP kinase, this makes perfect sense. Meaning, long story short, we can now investigate effects after enriching medium spiny neurons that were masked by contaminating cells before. So we can now address scientific questions that we were not able to address before. So in summary, my experience so far with the FACS sorter, which I used from 2013 to 2022, was like, yeah, well, I spent almost 1 year to develop this protocol. Then one disadvantage of this sorter is its giant size and requires a whole room. The sorting itself is quite cheap, but the maintenance of this machine is very expensive, and we have an average of, I would say, 5 to 8 severe malfunctions per year, and the service technicians are hardly available. So sometimes it takes them, especially during Corona, 1 to 3 months to perform service maintenance or assist with any malfunction. Another experience I had was time and time again that any service or maintenance changes my saved setups. So pressure, speed, scan setup. is then not comparable anymore, and I have to adjust the settings time and time again. So in my eyes, my experience is it's highly variable, insecure in terms of contamination, and it's unstable, which might not be a problem for cell lines, but for neurons it is. Now recently we started to work with the MACSQuant Tyto since 2021, and indeed it just took me 1 month to adjust the protocol and obtain living and healthy cultures. And also, I recently taught a master student to set up these cultures, and it took her 14 days to learn the technique, and she didn't have any contamination or problems with the sorter so far. Big advantage, it's benchtop size. Big disadvantage, the sort itself is quite expensive due to the material, but it does not really require maintenance. We only had one malfunction that was due to a software problem, but the service technicians were available within at least 3 to 7 days. And the only service we had was the installation of a new software. We didn't have any other problems, and it's running super stable. So I have to say the variability is quite low. Sorting itself is secure and sterile. It's very stable, and I have to say so far it's impressively reproducible. So in summary, for the direct pathway medium spiny neurons, we obtained a yield or concentration of 50 to 60% in the FAC sorting and 70 to 80% for indirect pathway medium spiny neurons. One of the biggest disadvantages of the Tyto sorter, we cannot sort red fluorescent cells because it's not equipped so far with a yellow laser that is able to detect fluorescent dyes at 550 nanometers, but it's able to sort GFP, and we end up with a comparable concentration between 60 and 90%, which is absolutely comparable to the FAC sorter. And in comparison, that's what a non-sorted culture looked like back in the days and after FAC sorting, and that's what a non-sorted culture looks like today and after Tyto sorting. So absolutely comparable. The only thing I have to admit is that the sorted cells from the Tyto sorter look much more healthy. So this is a pretty dense culture of indirect pathway medium spiny neurons after Tyto sorting, and as you can see, there is actually not really an astrocyte in this culture. It's 7 days in vitro and looks already quite good. And the big difference is, so first of all, in the FAC sorter, the pressure is quite high. We have electric charges, we have a high plate voltage that is all dangerous to our cells. Still, of course, we have the laser, and to my experience, I can sort 700 microliters in 30 minutes. The contamination risk is quite high. The reproducibility is moderate, service is very expensive, sorting costs are moderate to low. And we can sort blue, green, red, and far red. Now, the Tyto sorter uses only moderate pressure. It does not use any electric charges, which is the most important point. It does not use any plate voltage. Still, we have the laser, of course, but also the time is much faster. So we can here sort 2000 microliters in 30 minutes. The contamination risk is very low. Reproducibility is high. Service costs are so far low, but the sorting cartridges are still quite expensive. And we cannot sort red cells, but all the other dyes. So again, I want to point out marker specificity. I said this before, it depends on how specific your marker expression is, and this determines actually the purity of your cultures. And with this, I would like to thank the members of our institute. First of all, my primary supervisor, Professor Michael Sentner, who gave me the chance and supported me in developing those protocols and set up these protocols in the FAC sorting technique. I would like to thank our technicians who supported me, as well as the members of our animal facility. I would like to thank the Department for Microbiology and Professor Rudel, where I'm performing the FAX sorts using the BD FAX ARIA 3 with the direct pathway medium spiny neurons. And I would like to thank the group of Michael Hulicek. Together with them, we are using the MACSQuant Tyto sorter that I'm using for the indirect pathway medium spiny neurons. And of course, I would like to thank the members of Miltenyi Biotec, who are always very supportive and helping with all technical devices, issues, questions, and so on. And with this, I would like to thank you for your attention. And if you have questions, then I'm happy to answer them.